Ah.
And multiple sequence alignments.
Bootstrapping and multiple sequence alignments. Okay. So what's a multiple sequence alignment? What topic was this under?
Phylogenetics.
Right, so this is phylogenetics. So we can talk about bootstrapping and we can talk about multiple sequence alignments. Which order do those two come in?
The alignment and the bootstrap.
Alignment first.
Are they related?
They are somewhat related. So you align first and then you bootstrap. So an important principle here is that you can build, as you know already, you can build a phylogenetic tree based on any set of traits that you can observe in an organism. So that could be morphological traits, it could be behavioral traits, how they look, how they act, or it could be DNA sequence. So the sequence alignment part only has to do with phylogenetic trees that you're constructing based on DNA sequence. Bootstrapping is something you can do with any sort of tree, regardless of what traits it's built on: DNA, morphology, physiology, behavior, whatever. So bootstrapping is not specific to DNA sequence alignments, in other words. But if you were going to build a DNA sequence-based tree, you'd have to do the alignment first, then you'd bootstrap. So we have up with some sort of set of sequences. So let's say we've got 3 species. Species 1, species 2, and species 3. Like this. So this is multiple sequence alignment. So here are 3 sequences that we got from 3 different species. Species 1, 2, and 3. So we need to figure out, this is basically 10 different traits. Well, oh, it is, good, 10. 10 nucleotides from every one of the 3 species. Where we consider each nucleotide, right, each letter of each position in a chromosome to be a different trait. The first thing we have to do is figure out which of these traits are homologous. Right, so critical to know that you're comparing apples to apples, literally, when you're talking about traits. So you have to be comparing, you don't want analogies, you want homologies. So how do we align these sequences? These are all related sequences, say they're from all the same gene. Do they look like they're the same sequence? Do they look like these sequences are related at all? Not at first glance. If they were related, what would you expect to see?
Same sequence?
What?
The same sequence?
The same sequence, or in closely related species, would it be the identical sequence or would it be closely related sequences? So there'd be some genetic variation between them, but they'd mostly be the same. And that's how we would know we were looking at the same gene or the same region of the same chromosome. That's how we would convince ourselves that we were looking at homologous traits. It's the same gene, different species. So that's where the multiple sequence alignment comes in. When you sequence DNA, you have to figure out which parts of the sequence are related to which parts of the sequences from different organisms. So are there any sequences in these 3, are there any bits of sequence in these 3 sequences that are shared? So the way I drew it, we've got AGA, CATA in that first sequence. Looks like it's related to AGA, CTTA. So each of these characters, the A's, then the G's, the A's, the C's, here's a mutation. There's an A versus a T. Here's a T and a T, here's an A and an A. So all this means is these are the parts of sequence from the same region in the genome in these two organisms, two species. It's just that the DNA sequences, one has a little bit of extra on one end, the other sequence has a little bit of extra sequence on the other end. So to align these, what you would do is move this sequence over 3 nucleotides. Then each of those nucleotides that are matching would actually be literally on top of each other. So that would look something like, and this is what computers do when they perform multiple sequence alignments. So then you would line up this next sequence, AGACTTA, and that sequence would continue off the screen to the right, the GAC that I haven't written. So now these two sequences are aligned. There would be 3 hyphens there indicating we don't have sequence in that part of the gene for the second species. And then what we do for the third sequence. Does it have something that looks like AGAC8TA? This part that seems to be shared. So clean that up a little bit. So how could we align that third sequence? So maybe it's not related at all. And you'd have to be able to distinguish this. So you need to know when you're trying to align sequences that don't actually align. So that third sequence, that third species, we might have a sequence that has nothing to do, it's not from the same gene, for example, as the first 2 sequences.
So 1 and 2 are related?
So you'd conclude that 1 and 2 are related sequences because they share a certain amount of sequence in common.
Is there a certain length of nucleotides that confer that?
Yes. For you, no, but the answer is yes. So the simple answer, yes, there is a minimum length of sequence you'd want to have in common to be fairly statistically certain that those are not by chance.
And what length is that?
Okay, plug your ears if you don't want to know the answer to this, because it's not going to be on the final. So how common is every nucleotide? What's the probability that if you look anywhere in the genome you find an A? 25%. So a length, so if you wanted to know, is this 7 nucleotides, AGACTTA, for example, is that 7 nucleotides unique or not? The probability of getting that sequence is 1 out of 4 for every one of those 7 nucleotides, or 1 over 4 ... which is 1 over 4 to the 7th. That's the frequency with which you'd expect to see any 7 nucleotides of any sequence. So you pick the 7 nucleotides, that's how frequent it should be. Then all you need to know is how big is the genome, how many nucleotides are there in this genome of the sequence you're looking at. And then you divide and you figure out what's the likelihood, how many times will that 7 nucleotide sequence by chance show up in somebody's genome. Okay, now you can unplug your ears if you were plugging your ears. So that's in truth what we do when we're looking at how unique is a sequence. So was I trying to be too tricky when I wrote that third sequence that I just totally confused myself? I guess so. Because I agree, I don't see anything that looks anything like that. Oh, wait. Wait a second.
I think the T's just changed from A.
There we go. So circled in red would be a way to align the first and the third sequences with each other. It's with one mutation, where that A has become a G in the other species. So in this case, for this example, say we're moving on now to ask about bootstrapping. how many characters do we have, or how many nucleotides do we have that we have from all three species at this point? It looks like just, let me switch colors again. Tell me if I'm right or not. I think that AGA, which is there in that sequence and there in that sequence. So if we did a multiple sequence alignment with all three, those three would be, that column of three nucleotides would be in the middle with some sequences sticking off in either direction. In other words, that's what that multiple sequence would look like. So we didn't get those last four nucleotides from species 1 or 2. We got those last four nucleotides in species 1 and 2, but not in species 3. And we didn't get the first three nucleotides from species 2. So we have that column of characters, once you align the sequences, that we could use to make a phylogenetic tree. Normally a computer does this for us. I might ask you to do something like this on an exam. And all you do is you look for what sequence is common between the three or more sequences and then draw them like that. With dashes for missing characters, missing information. Questions, concerns, additional clarifications on this? Being long-winded about this, but there's the sequence, the distance matrix for these three sequences and the phylogenetic tree. What's the branch length between? Right. There's one difference between species 1 and species 3 and species 2 and species 3. Bootstrapping. We want to know whether or not this is the correct tree. So what is bootstrapping?
You run it multiple times, the same thing, and then you get like the average of
Right, so what we,
probability.
yeah, what we want to do in bootstrapping
Replacing one character state with another.
is randomly pick traits from this initial sequence alignment in this case. So we got 3 traits, so this will be pretty easy. We want to make new sequence alignments made of randomly chosen sets of sequences. So for example, if we number these positions 1, 2, and 3, which is hopefully not going to be too confusing because those are the names of the species also. What we want to do is come up with a number of sets of data that are random combinations of column 1, column 2, and column 3. So if I was going to do this on an exam, I would say, pick some character states. So we randomly pick sets of numbers, 1, 2, and 2, for example, 3, 1, and 1, for example. Just random columns. And then you'd write out those sequence alignments as well. So column 1 was AAA, column 2 was GGG, column 3 was AAG, and again, column 1, twice. So the critical point here is that we've made new modified sequence alignments that has the exact same amount of information as the original one. So 3 columns of data, 3 traits, 3 characteristics. They're just randomly chosen. Never, well, it could be by chance 1, 2, 3 again. You do this, so now we've got 3 different multiple sequence alignments. So now what we're going to do is we're going to draw the trees for the next 2, the ones we just made. So this was tree Roman numeral 1. What is the second tree? So there's alignment 2, alignment 3. So alignment 2, what is it going to look like? What's the distance? 0. All 3 of the sequences are identical. So what does that tree look like? There's not even a branch. It's just a dot. Or nothing. You have dot, leave it blank, whatever. So they're identical. What about tree 3?
But there is no 3, so there is no 3, it's just 1, 2, 2.
So I'm sorry, these are species. I was afraid this was going to get confusing. So these are species 1, 2, and 3, not columns 1, 2, and 3. Should I rename? Let's see. Columns little a, little b, little c. How about that? So we have 3 columns of data.
So even though a species is not in the tree, you would say included.
My apologies. Hold on just a second. This is why I record this, so you can enjoy all of my mistakes at some point in the future. Okay, so sorry, ask that question again.
Even though you're not included in there, you're still included in the tree. Why?
Even though I include
3. Species 3.
Yes.
Not in one of those.
So the species, so each one of the species is represented in each one of the trees. It's each of these 3 columns of information that are not necessarily present in all of the trees. So in every one of these multiple sequence alignments, we have species 1 sequence, species 2 sequence, species 3 sequence. So we have data from each of the 3 species represented in every bootstrap replicate or every version of the multiple sequence alignment. What we don't have in this case, for example, is we don't have column B of the sequence. And that's by chance, because when a computer does the bootstrapping, it randomly picks, in this case, 3 columns. So it might by chance not pick column B. It might instead pick column A twice. You could have a bootstrap replicate where it's the same column 3 times. It could be BBB, or AAA, or CCC. And the point is, again, just that the better the sequence alignment is, or the more informative the sequence alignment is, the more times you'll get the same tree over and over and over again, regardless of which set of data you're looking at from your original sequence alignment. Okay. So for the Roman numeral 3 tree, this one, what's it going to look like? So 1 and 2 are identical sequences. So it's going to look the same as tree 1. There's one difference between species 1 and species 3, but you're right, no difference between species 1 and species 2. So it's going to look exactly the same as the first tree, the branch length of 1. So the very end of the bootstrapping process is you count up how many trees you've drawn. In this case, we've done 3 trees, and you ask how many of them have the same structure. Right. So in this case, trees 1 and 3 are identical. So 2 out of the 3 trees, or 66% of the trees, have this shape. So then you would write that as the percentage, 66. Usually at a node, but this tree doesn't have any nodes, so on the branch, distinguishing the 2 species. And that tells the viewer that 66% of the time when you did bootstrapping, you got this tree. And that's what tells you how confident you are that this is the tree that your data most strongly support. Likewise, I guess, this tree at the dot or at the nothing, you could write 33. 33% of the time that was the tree that your data suggested.
After all,
Yeah.
I don't understand why you repeated a sequence, so like A, B, B, and C, and A. I don't understand.
So when you bootstrap, you randomly pick columns of information from the original matrix of characters, whether it be DNA sequence or traits or whatever. And it doesn't have to, if you picked this process of choosing which columns is random, or as random as a computer can be, which is not, but it's random enough, and occurs with replication, which means you take the 3 columns in this case and the computer picks A, B, or C. And then it picks again, A, B, or C. And then it picks again, A, B, or C. Which means that the computer could by chance pick A, or it could pick A, B, B, or it could pick A, B, C, or C, A, B, or any combination. What would happen, everybody, what would happen if bootstrapping occurred without replication? That is, if once a computer picked a column, it disappeared from the pool of columns of data to choose from. What would you get every time if you had to choose 3 columns of data and there was no replication?
It would always be the same one.
Yeah, it would always be the same 3 traits. You'd always get the computer picking A, B, C, A, B, C, A, B, C, and that wouldn't tell you anything. Because you'd be looking at the same set of data every time you drew a tree. So it has to occur with replication. That is, when you pick a column, it goes back into the pool that the computer can pick from in the next round of randomly selecting columns of data. And of course, most phylogenetic trees are not built on 3 columns or 3 traits. They're built on, in DNA sequence, hundreds, thousands, tens of thousands, in which case this becomes a lot less stupid and a lot more useful. Because then you get truly different looking trees a lot of the time when you do this subsampling, the random selection of traits.
I'll go again. Directional selection.
All right, directional selection. What sorts of directional selection did we talk about? If I remember correctly, there was a great slide with a lot of different graphs of what allele frequencies would do under different types of selection. Pardon? This wasn't when we were talking about drift. It was close to that. Do you know what date that was? I'll pull up
Yeah, it was,
the slide.
160314.
Okay, 314. Thanks.
We talked about, allele frequencies going into fixation in relation to being dominant, recessive, or codominant. It's slide number 5.
Okay. Here we go. Okay. So directional selection, briefly, is when an allele goes from a low frequency to in the other direction. So it starts low, like down here. A new mutation arises. The y-axis, in this case, is allele frequency. And directional selection means it's going from nothing to fixation. So it's being selected for or it's selected against. It's not one of the more complicated cases of selection. Then this is a really good thing to discuss. So why is it that a recessive allele has the shape shown there where it takes a really long time to start increasing in frequency? So we've got a new mutation in a population. It's really rare for a long time. Then it becomes common. Pardon?
Maybe the environment changed to allow it.
Was something about the population. So it could be that the environment changes and now that mutation is advantageous. Let's say it was advantageous from the start, but it's recessive.
Right, so you need 2 of them in order to actually show the phenotype.
Right. So the question is, what's the probability when you have just one, say, starting out, you have one individual in the population that has that one mutation. So they're heterozygous for a beneficial recessive mutation. You have to be homozygous, you have to have 2 copies of that allele to see the benefit. So this really long lag time is just that individual introduces that new allele to more members of the population in subsequent generations, and generation after generation, it takes a long time until finally some individuals in that population by chance have 2 parents that were heterozygous for that mutation that mate, and now you get homozygous recessive individuals, and they start experiencing the fitness benefit. And as soon as you get some homozygous recessive individuals in the population, then when individuals start seeing the benefit of being homozygous, that mutation goes to fixation quickly. So that 600 generation or so lag time here just has to do with the fact that it takes a while for that mutation to spread slowly into more and more individuals in the population. And those heterozygotes, say it's a big A over little a, let's say little a is the new mutation, those heterozygotes, big A over little a, don't get any fitness benefit as heterozygotes because it's recessive. The other thing we talked about is why dominant mutation and codominant mutations look slightly different. The codominant mutations reach fixation slightly faster than dominant mutations. And we talked briefly about why that is. So why does, as it says here, when A is dominant, its initial increase in frequency is most rapid, so it's got the steepest peak. When the new allele, big A in this case, is dominant, it goes to fixation, has a slightly faster slope. It starts increasing in the population more rapidly, but then it slows down. It takes a long time for big A to be the only allele in the population.
Because even the heterozygous is going to have the detrimental allele otherwise.
Right. So it's the case here is that in dominance, when you're heterozygous, big A over little a, there's no way to get rid of the little a allele because you see the full effect of the dominant allele even when you're heterozygote. So it's basically impossible to get rid of all of the little a's in the population because they don't affect your fitness. So if you're heterozygote, you're just as fit as a homozygote. So if you had a big A over little a, if you're heterozygote, you have the same fitness as being homozygous for big A. There's no benefit of getting rid of the little a's, basically, in the population. And in codominance, why does codominance help? What's the concept of codominance?
Blending inheritance.
It's kind of blending inheritance. So a heterozygote would sort of be 50%, have 50% of the advantage of a homozygous big A. And so there is a disadvantage in codominance to being heterozygote. You're better off than being homozygous little a, but you're not quite as good as being homozygous for the capital A allele, the new allele in this case. It's a minor point, but that's what we talked about on this slide.
I'm sorry, can you explain one more time why it's better to be dominant?
So in a case of codominance, flip back. So in codominance, let's say capital A is the new fancy good allele, the better allele, the most fit allele. So under codominance, what's the relative fitness of these three genotypes? Which one's best? Big A, big A. Little a is going to be the worst, and big A over little a will be, good grief, intermediate is what I'm trying to spell. So that means that there's some way to select against heterozygotes because they don't have the best fitness. They're intermediate. So that's codominance. What about the case of simple dominance?
It's similar.
So who's the best? Big A, big A. Who's also the best?
Big A, little. Big A, little.
And then the original genotype, little a over little a, is still the worst. So this is the critical difference between simple dominance and codominance, is that in simple dominance on the bottom, there's no way to select against heterozygotes. So that's why it takes so long for the population to get rid of little a's, because how do you eliminate little a's from a population in this scenario? What has to happen? How do you get rid of the last one little a genotype in the whole population? How does big A actually achieve fixation? Nobody left in the population has a little a.
It has to die because of other reasons, like
So you wait for a chance event, as you're suggesting, like the individual happens not to reproduce, drift, or they die before they reproduce or something like that. Or you have to get to the situation where if you have two heterozygotes mating together, right, this is why you can't get rid of the little a's. Every time heterozygotes mate with each other, they produce some that get selected against, but they maintain the little a allele in the population. Even if it's matings of, once the big A allele starts getting close to being in 100% of the population, when there are still heterozygotes around, you still have those little A alleles floating around that can't be selected against because those individuals have the same fitness as the homozygous big A's. So indeed, you have to wait for chance events to kill all of the individuals that have the little a's, or at least prevent them from mating. Do you want to go on and talk about overdominance and underdominance, or those are the other two types of selection that we talked about? Or we could move on to a different.
Could you talk about over and under dominance?
Okay, so over and underdominance. So again, to remind you what this looked like back in the day, this was the slide on overdominance. Heterozygote advantage. It's like when you cross two different races of wheat together and you get hybrids that thrive much better than either parent. So there are cases like this in nature where being a hybrid, being the offspring of two genetically different individuals, is actually more beneficial. And the reason that this is called overdominance is, as it shows here, the heterozygote has higher fitness. The A1, A2 on the left here, it's got the highest fitness. So what happens in this population is you never get rid of, this is not directional, you never see either complete fixation of an allele or total loss of an allele. What you wind up seeing over time is the two alleles in the population reach some sort of balance, an equilibrium. So as it shows here, after an infinite number of generations, you wind up getting something like a little over 30% of the big A1 allele in the population. And that's because there's a benefit to being heterozygous. So there's no benefit, evolution doesn't work to get rid of one allele. It maintains both, because the best genotype, the most fit genotype is the heterozygote.
So A2 would have a similar graph.
So what would A2 look like? And why? So let me draw this out for you, and I'll flip back over here. Bless you. Oops. So if allele A1 starts, so this is time, generations, whatever. If it starts high, it'll dip down to about 30%. If it starts at 0, you'll see it increase in frequency to about the same equilibrium, about 30%. So what would a graph, the question was, what would a graph of allele 2 look like? Right, so if there are 2 alleles in the population and this frequency reaches 30%, then what's the frequency of allele 2 at equilibrium? It's got to be 70%, the difference between 100% and 30%. So if we drew that in red, so in the case where allele 1 starts at 0% and increases to 30%, allele 2 would start at 100% and would go down to 70%.
But it has to. But wouldn't that increase the number of A2 individuals because it has higher frequency?
Right, so
That's
this is,
not an equilibrium.
so this is an important point about this sort of selection. Why is it that both allele 1 and allele 2 don't achieve 50% frequency? So why don't we see that? Why is it 30-70 instead of 50-50? There's something subtle about this graph down here that's important. The fitness of individuals that are homozygous for allele 2 is a little bit higher than the individuals that are homozygous for allele 1. Which means it's better to be 2/2 than 1/1. So that's what pushes this frequency of allele 2 to be higher than 50-50. It's better, there are more allele 2s in the population because it's more advantageous to be homozygous for allele 2. And that's why the frequency of allele 1 at equilibrium is only 30%. It's worse to be 1/1 than it is to be 2/2.
And it's better to be, well, A1 and E.
It is best to be a heterozygote. Yeah. And so you might be imagining, or you might be totally blanking out and glazing over right now, you might be imagining that the math behind this is a little bit more complicated to try to guess based on, you'd have to know how fit relative to one another the 3 different genotypes are, and that predicts and that creates exactly this plot.
According to the numbers, shouldn't A2 be more beneficial than A1 A2 though?
No, this is the case of, only because this is a case of heterozygote advantage. So we go out into nature, we discover that the heterozygous genotype is the most fit. And that's what this shows. It's most beneficial to be allele 1/allele 2. And that's just because of whatever the actual example is.
So then,
That's why this is not directional selection, in which case you would totally lose one or the other allele because having one allele would be best and the other one would be worst. In this case, you have to have both copies, both versions of the 2 different alleles to be the most fit. So there has to be maintenance of both of those alleles in the population to achieve that.
so allele 2 will be somewhere in between the 50 to 70% range.
So the frequency of allele 2 will equilibrate at 70% if what has been shown is that allele 1 equilibrates to 30%. The sum of those two has to equal 100%.
So if A1 is to increase, A2 has to decrease it in order to maintain the equilibrium.
At this point, this is the stable equilibrium. That forevermore in the population, you're going to have 70% allele 2s and 30% allele 1s. But what happened initially, yes, was that allele 1 had to increase in frequency to reach that stable equilibrium.
So this is still all overdominance, correct?
This is overdominance, heterozygote advantage. And the only thing I really want you to be able to do with this information is to be able to detect it. So if you see a plot like this, you don't have, of course, you don't have to be able to predict what the equilibrium frequency is going to be. We haven't talked about the math. I just want you to be able to look at this sort of a graph and say, okay, what this looks like is both of the alleles are being maintained in the population. Therefore, it's not directional selection. It's not fixation or loss. Both of the alleles get saved in the population. That suggests that there's a benefit to having both alleles in the population, which probably means there's heterozygote advantage.
What kind of selection is occurring in underdominance?
The opposite, which is a stupid thing for me to say. So here's it, right? If overdominance is heterozygote advantage, then underdominance is heterozygote disadvantage. That is, you know, you're not supposed to mate with your siblings and brothers and sisters and cousins and stuff. That's sort of the idea of heterozygote disadvantage. And sometimes it's bad to do that.
So what type of selection? Is it like negative selection, like purifying selection, anything like that, or no?
No. So purifying, positive selection and purifying selection is something we could also talk about if there were time. But those have to do with, those are separate from overdominance, underdominance. So and the only thing to take away from this again is the idea that in this case, an individual who's heterozygous allele 1 over allele 2, for example, in the middle column, in this case has less fitness than either homozygote. So if I just told you that, without looking at the, don't look at the graph, everybody close your eyes or look down. What would you predict the graph would look like if I tell you that the least fit individual has both alleles? What should the graph look like? In the last case, the best fit allele, the best fit individual is one that had both alleles. So we see both alleles being maintained in the population. So what's going to happen if being heterozygote is the worst thing you could be?
So it's going straight and then separate?
It'll either go up,
Or
or depending on which allele you're looking at, it'll be fixed or it'll be lost. Yep. Okay, now you can look at the graph. That's what you see. But it's not, this, critically, last thing I'll say about this doesn't count as directional selection. Because whether or not, in directional selection, you always know that one allele, say allele 1, is better than the other. So you know it's, the direction means it's always going to go in one direction. This doesn't count as directional selection because it's always the most common allele that will fix. So you can't say for sure it's allele 1 that will fix. It just happens to be, and that's what that dashed line represents. If the new allele is common, it's the one that will be fixed and the rare one will be lost. Again, just to avoid having any heterozygotes in the population. But if it's flipped, if in this graph up here, if allele 2 happened to be the one that started out being more frequent and allele 1 was less frequent, then you'll see the opposite. So again, when you see, when it looks like in the population the alleles are, one allele is getting rid of, being gotten rid of, and the other one's being fixed, that could be an indication that there's heterozygote disadvantage.
I'm confused with how they start at that point, because that's not 100% of the alleles, right?
Right, so what we're looking at here is just one, allele 1. So they haven't, again, they haven't plotted, so this is, we're not looking at allele 2's frequency. So what might be
Would it
a real,
go up that way though, if it's at 0.4, which is less than half?
it depends on which way A1 actually went. The A2 will go the opposite.
right, indeed. So we're looking at A1, so if we're looking at the plot on the top, A1 starts at like 35% and goes to fixation. That means the allele A2 would start at 65% and then, yeah, go down to 0%. Yeah.
Will there be a study guide?
No study guide. So the new information, there's not going to be a study guide for. So that's what? Sex determination, speciation, sexual selection. The new material. And I'll post more details as I get closer to writing the exam. I'll post the usual details that I posted on about the midterm. Types of questions, relative point values, those sorts of things. And I'll try to tell you explicitly what the balance between old material and fresh material since the midterm will be. All right. So I've got office hours today from noon to 1:30. I'll be around some tomorrow and Friday, so stop by. I have some meetings that have been scheduled, unfortunately. So if you want to stop by during consultation days, you can, of course. And that's it. So one more question. No, there won't be any group portion on this.